Okay, this is, hold on. Away. Okay. Can't hear anything on here, so that's always a good sign. Yeah, I don't get sound. Whatever. We'll just have to play it through the computer. Let's do the Kahoot for chapter 20 before we get into our lecture on chapter 21. So let's get going, make sure everybody's signed in. I don't even know if people can even hear me talking on the Zoom. Is it even like it's not even there? It is registering. Okay. They changed my computer for those of you that weren't in the classroom. They literally replaced my computer, so I'm having to like reset everything. Bear with me. Thought we could maybe just finish the semester everything the way it was, but no. All right, so let's get going with our Kahoot. So I can see it's the weird angle. All right, what sort of organisms make up the normal biota of the nervous system? Okay, so the correct answer for this one is going to be none of these. The nervous system, as well as what we're talking about today, the cardiovascular and the lymphatic systems, they don't have a biota. They're considered sterile when it comes to microbes. The next question. What is the term that refers to the significantly decreased and compartmentalized immune response in the nervous system? There's a specific term that I'm looking for here. We talked about it with the eye as well. Righty, so this is called immune privilege. That's the term for that. It is a lowered immune response, but the term for it is immune privilege. Next question. Which types of organism can cause meningitis? My dog's rolled in something that smells like skunk, and I feel like I can smell it on me. So if you think I smell like a skunk, you are probably not wrong. So, so I apologize for that because I was just like, they smelled like it at home. I didn't realize that I might smell like it until I was standing up here. All right, so this is everything except for the prions. Most of us got this one. So we talked first about the bacterial meningitis, then we talked about the fungal meningitis, and then viruses can also be associated with meningitis as well. Typically a faster, improvement from those diseases though. All right, so which pathogen causes the deadliest form of meningitis? 3 of these are pathogens associated with meningitis. They're all bacteria. Which of these is the worst? The correct answer for this one is Neisseria meningitidis. This one causes the worst version of it, with the petechial rash and a higher mortality rate as well. Right, what type of illness does Naegleria fowleri cause? Naegleria fowleri is associated with amoebic meningoencephalitis. The one that Naegleria fowleri causes is the PAM version, which is primary amoebic meningoencephalitis. The granulomatous one, does anybody remember what causes that one? Acanthamoeba, exactly. So good job with that. Let's move on. Encephalitis, I'm assuming, is the next part. What type of pathogen typically causes encephalitis? One of the most common causes of encephalitis. This is going to be arboviruses. These are the ones that are transmitted by biting insects. Can you have encephalitis caused by these other guys? Potentially, yes, but it's more commonly associated with the arboviruses here. True or false? Thanks to the polio vaccine, polio has been eradicated from the planet. True or false? This one is false. We still have cases appearing in Afghanistan and Pakistan, where it's been hard to get the vaccine efforts to the rural areas and war-torn areas in those countries. Our next question, what kind of pathogen causes Creutzfeldt-Jakob disease, or CJD? So this is going to be prions. Prions are associated with spongiform encephalopathies. One of those is Creutzfeldt-Jakob disease. And the misfolded proteins will build up and form holes in the brain when they aggregate together. What is the mortality rate for rabies if it is left untreated? I feel like I've said it a lot. I won't take it personally if you get it wrong, but you might want to, might want to remember it. No pressure or anything. All right, so this one is 100% fatality rate if it is not treated. And by treatment, we mean, of course, getting vaccinations, shots, or even immunoglobulin within, respectable range from being exposed to a potential rabid animal before signs or symptoms appear. Once signs and symptoms appear, we're talking 100% fatality rate, no matter what. So which toxin causes spastic paralysis? We talked at the end of chapter 20 about two diseases that cause paralysis. One of them caused spastic, and one of them caused flaccid paralysis. Alrighty, so this one is tetanospasmin. This is associated with tetanus. Tetanus is the disease where the muscles can't relax. They become contracted and are unable to relax. Whereas botulism was the disease where we had botulinum toxin blocked the release of acetylcholine, which is the signaling molecule for contraction of muscles. And so flaccid paralysis is with botulism, spastic paralysis is with tetanus, and the name of the toxin with that is tetanospasmin. Our little podium, and then we'll get into the lecture. All right, beautiful. Great job, everybody. So let's get out of this and jump into our lecture for today. And just so that you guys are aware, our lab should not take super long day. It should be relatively quick, but you're welcome to stay behind and verify any of your results with me and check on that stuff if you would like to. At the end of lab, everybody should know what their organisms are for their gram-positive and gram-negative species. So double check that with me before you guys leave. Yeah. All righty. So let's get into it. Chapter 21, we're covering the diseases of the cardiovascular and lymphatic systems. We'll start off with the cardiovascular system. I'm not necessarily going to test you on any of the anatomy, but it just kind of helps to know what's going on with this. So in the cardiovascular system, blood is going to pump within a closed system, which is actually pretty important for this because we don't have a biome in here, but we have plenty of nutrients and oxygen and growth factors and things like that in the bloodstream. It is an open stadium with tons of open stadium seats for any pathogen to come and take advantage of. And so that is something that would not work to our advantage if a pathogen did get in there. So we keep it as a closed system to prevent pathogens from gaining access. Of course, there could be breakage in that closed system, and that could potentially introduce pathogens into there. Blood is pumped by the heart muscle contracting. The different chambers of the heart help to push the blood throughout the body. Then the blood vessels are carrying the blood throughout the different tissues of the body. providing the tissues with oxygen and nutrients and carrying away waste products like carbon dioxide. Blood vessels that carry blood away from the heart are called arteries. And then we have veins that are carrying the blood back to the heart. And that's really how they're going to be defined, is where the blood is moving to or from the heart. And then capillaries are sort of connecting the veins and the arteries at the smaller level, and also provide that gas exchange within the tissues. The lymphatic system we learned about previously in chapter 15, but this again, just to summarize, it's going to be all of our immune cells and the tissues involved in the immune system. This is the lymphatic vessels, as well as specified lymphatic tissues like the lymph nodes. And the spleen also is going to be considered in both of these categories. So the spleen filters out the damaged and old red blood cells, as well as filtering out the blood for pathogens. So the spleen filters the blood itself, whereas our lymph nodes are filtering the lymph, that fluid that's coming back out of the extremities before it rejoins the circulatory system. So for our defenses in the cardiovascular system, pretty much like we said, it's a closed system and it's a whole bunch of open stadium seats. But the good thing about the cardiovascular and lymphatic systems is that it's highly populated by our white blood cells. And these are our immune soldiers, right? And so those immune cells are always circulating within our immune system. So we've got thousands and thousands of soldiers guarding those open stadium seats, so as we could reduce the likelihood of infection happening. Anytime that there is a condition of the blood, we call it anemia. So when we say anemia, which is what I was trying to avoid saying directly, so anemia means an without, emia, blood. So you're low on your red blood cells. So that's why anemia is that way. Viremia, these would include viruses that can cause meningitis. They're usually going to be found in the bloodstream as well once they are infecting the meninges and causing inflammation there. And so viremia refers to viruses in the blood, fungemia, fungi in the blood, and bacteremia is bacteria in the blood. When pathogens, and especially bacteria, specifically, this is where most of the threat is going to come from, when they're actually multiplying in the blood, not just being carried around by the blood. If they're being just carried around by the blood and they're there, then that would be bacteremia. But if they're actually multiplying in the blood, that's septicemia. So the blood has become septic. And so when we talk about asepsis and performing things with aseptic technique, preventing the contamination, once it has been contaminated and these guys are flourishing and growing in the bloodstream, and think of it as sterile media that's become inoculated, right? And those guys will multiply in there. And once that happens and the infection is established in the actual bloodstream, that's septicemia. And so the cardiovascular and lymphatic systems are closed, just like the nervous system, and are protected from invasions of pathogens. The only way they're going to get in is when there's some sort of break in that barrier, potentially. You can have microbes floating in those areas transiently, so they should be cleared out by the immune system. But again, if they actually end up multiplying in the bloodstream, then that is going to be septicemia. We're going to start with our diseases. Our first one is going to be COVID-19. We think of COVID as being a respiratory disease, but it in fact can multiply in most of the cells of the body. This is caused by the virus SARS-CoV-2. So SARS-CoV-1 is associated with the SARS disease, which is severe acute respiratory syndrome. There's an outbreak of that in the early 2000s in China and in other countries in Asia. There's a huge concern that it might cross over into the United States because it did seem to be a fairly deadly respiratory infection, but it honestly was nothing compared to what COVID ended up being with this different virus. There's a lot of speculation about where COVID, the virus SARS-CoV-2 that led to the disease of COVID, where that actually came from. But most of the evidence now points to it was being worked on in a research lab in China, potentially to work on bioterrorism agents, though, that's only speculation. And so that got out from that environment and potentially was spread to people from that. SARS is mostly primarily respiratory infection, whereas COVID is going to be more systemic. And there's reasons for that. But the type of virus that's involved in this is called the coronavirus, since that's why it got its name as the COVID coronavirus infectious disease. SARS-CoV-2, the virus that's associated with COVID, infects more than just humans. And there's a lot of different coronaviruses that cause a lot of different diseases. Most of them are associated with like colds and things like that, but some of them are more serious illness. These guys are able to enter cells through this ACE2 protein. I won't test you on that, but it's a protein marker on most of our body cells. including the lungs. Of course, it's where we get the respiratory aspect of it, but it also can attach and infect brain tissue, heart tissue, blood vessels, kidneys, liver. It can infect the placental tissue itself and the gastrointestinal tract as well. And it is spread through, it is airborne through respiratory droplets, and that's how it's spread, but then it's able to disseminate throughout the body thanks to the circulatory system. Mutations have given rise to certain variants of the SARS-CoV-2 virus. The original version was considered very virulent, but each new mutation seems to be more contagious and more virulent. However, when we look back on this, we look at people that had the original COVID infections, that they were being hospitalized and things like that. And then these later versions of the different variants, though they are actually more transmissible and more virulent, cause worse disease, we don't see that as much. And we're out here still talking with each other and communicating with each other and not dying from COVID. And there's a reason for that, right? We're going to talk more about that in a minute. So the COVID infection, we start off with prominent respiratory symptoms, and the infection can actually set off a cytokine storm. So what happens is people who have get COVID and have never had the vaccine or anything like that, their immune system gets triggered to over-respond to it. And that's what causes the deadly effects. Instead, with the vaccine, we can train our immune system to respond the right way to the virus instead of overreacting. So the vaccine has pretty much led to a significant decrease in the number of cases that have cytokine storm. And the cytokine storm, that overreaction, the inflammatory response, is what led to the deadly effects of COVID. It wasn't the virus causing the deadly effects, it was the immune system's response to the virus. And so now we've been able to regulate that by introducing the antigen through the vaccine and hopefully triggering a more appropriate immune response as opposed to having the cytokine storm effect. And the cytokine storm would eventually lead to things like rashes, muscle pain, heart damage, joint pain, and blood clots. And then these would have lasting effects. And it also is a neurotropic virus that can infect the nervous system as well. So then that could lead to loss of the taste and smell, affecting the actual nervous system there, headaches, and long-term cognitive decline and dementia as a result of the infection, and usually in response to the inflammatory response associated with the virus as well. Up to a third of people that are infected will be asymptomatic, a third. And that's pretty impressive when we're thinking about people going out with masks and social distancing and things like that, that some of those people were out there thinking, I don't want to get COVID, but they might have actually had COVID and been spreading it around to people, not knowing that. So it's pretty useful information now knowing that. With masks and social distancing and hand washing, we could prevent the spread to some extent, but don't attribute the success of our being able to be in the room right now with each other without masks and things like that to the social distancing and the masks. Attribute the success of that to the vaccines. The vaccines are what has retrained our body to not have that cytokine storm effect, and so now it's safer to have the infection. And so hasn't necessarily prevented the spread of the infection, but it has made it safer to have the infection. Now it's more of a cold or flu-like response as opposed to the cytokine storm effect that was putting people in the hospitals. We already talked about the COVID vaccine a little bit, but again, there's two different versions of it. The one that is literally like putting mRNA into your body, and then your body will translate it, put the spike proteins on your cells, and you have an immune response to that, which is what explains getting COVID arm like you would see in some people, which I got. I've never had any side effects to any vaccines in my entire life until I got the COVID vaccine, and I got the COVID arm, and my whole arm was red, and a lot of it was like swollen up, not massively, but it was definitely like swollen up from the inflammatory response to my cells expressing the COVID protein. But it did provide a better immune response so that when I got COVID later on, I didn't have to be in the hospital. I got 104.5-degree fever, but I imagine that if I hadn't had priming from my vaccine, that I likely would have wound up in the hospital as a result. So there's that. The Johnson & Johnson vaccine uses a different mechanism. It takes basically an adenovirus, which is just a cold virus, and we've hollowed it out. And so the only gene that it expresses is the spike protein for the COVID virus. So it's a different virus, and it just has on its spikes, instead of regular adenovirus spikes, it has COVID spikes. So we respond to that, create immunity to COVID that way, without all the other factors of the infection stimulating the immune response, leading to that, that cytokine storm. So there was medication called Paxlovid that was basically developed to treat COVID. So there are medications for treating more severe cases of COVID for people that aren't improving on their own. Moving on. Next is endocarditis. The endocardium is the inner lining of the heart, and so endocarditis is going to be inflammation of that inner lining of the heart. There are basically two forms of it, the acute and the subacute. Essentially what happens is you might have damaged heart valves from heart disease, high cholesterol, for diet, metabolic disorders or whatever, or you might have prosthetic heart valves. And so inflammation can trigger the likelihood of transient microbes taking up house in the inflamed tissue that's already damaged. And then that leads to infection in the inner lining of the heart. You can also have endocarditis develop from trauma and even circulating immune complexes getting stuck in these structures as well, then leading to the inflammation. Signs and symptoms of the acute and then the subacute. So the acute endocarditis, again, it's a shorter term that you'd be having the signs and symptoms, but there'll be stronger signs and symptoms compared to the subacute version, which is more chronic and it's long term, but it's less pronounced signs and symptoms. So with the acute versions, you have fever, anemia, abnormal heartbeat, symptoms of heart attack, shortness of breath, and chills. So you'll have those cardiovascular sorts of symptoms associated with the heart, as well as more widespread infection symptoms. We also have abdominal or side pain, Janeway's lesions, and Osler's nodes. These Janeway's lesions and Osler's nodes are basically a result of red blood cells clotting or rupturing capillaries. It's sort of like having the petechial rash, but it's usually localized to it gets stuck in like the nail beds or in like the soles of the feet and stuff like that. And we have these like reddish brown spots that will come out in like the nails and things like that as a result of that. The abdominal or side pain is relating to the work that your spleen is having to do with clearing out bloodstream infection, as well as damaged blood cells as a result of the inflammation. And then that gets into the subacute endocarditis, which again, similar symptoms, but just less pronounced. And then over time would lead to enlarged spleen, so splenomegaly, as a result, again, of responding to the damaged red blood cells, the inflammation, and the fact that there are microbes in the circulatory system. And then we have clubbed fingers and toes. And if you don't know what clubbed fingers or toes are, it's where you have like a rounded effect on the end of the finger. And instead of having the nail come out relatively straight, it'll be a larger nail bed and it'll be rounded out over the top of the nail. And that is never a normal thing. And so it's like most of the time, like this is not it's not like your hair is blonde and mine is brown kind of genetic effect. It is usually due to genetic defects in metabolism, lack of nutrients, and illness. And so if somebody has clubbed fingers or toes, it's always a sign of some sort of underlying illness going on. It could be something severe or something minor, but it's worth having a doctor check out, hey, I have clubbed fingers and toes. Let's do some blood work and try to figure that out. So that's not a normal effect of just being, genetically, physically different. It's usually a side effect of some sort of underlying disease. So moving on to sepsis. This is the septicemia. This is where we have the actual infection in the bloodstream from these microbes. So signs and symptoms associated with just having this infection of the bloodstream: fever, altered mental state. We're having issues of getting nutrients to the brain because the microbes might be impeding the flow of blood and oxygen to the brain tissue. You'll have shaking, chills, usually so bad that the muscles are getting involved because we have a systemic infection. It's not just local. It's not just in like the respiratory system. And then your immune response is floating around. The infection is floating around and the immune response is floating around with it. And so we have a much more pronounced display of signs and symptoms associated just with having infection. And then we have also gastrointestinal symptoms, again, because of the blood flow to the tissues there. Increased breathing rate, respiratory alkalosis, and low blood pressure resulting in loss of fluid from the vasculature is all basically in response to the fact that we're not able to get oxygenation and removal of toxins done effectively because the microbes are impeding our bloodstream from being able to do its job without interference from the inflammatory response. Right, so let's talk about some effects of the septicemia. So endotoxic shock is a pretty common one with gram-negative bacteria. Remember, endotoxin or lipopolysaccharide is found on the surface of gram-negative bacteria, and it is a PAMP. It is something that can trigger the inflammatory response, and it's a very strong trigger as well. And so this can lead to shock as a result of that, and this is a drastic loss of blood pressure and can also lead to lack of ability to get the oxygen to the extremities and to the brain, and we have results of that leading to basically widespread severe signs and symptoms associated with what is going on systemically from the inflammatory response. And this can lead to death very quickly as well. Gram-positive bacteria, they don't have endotoxin, but they can produce toxins that can elicit a similar response. And keep in mind that any presence of peptidoglycan would also be considered a PAMP and can trigger the inflammatory response as well. So it's usually going to require other toxins to trigger something similar to endotoxic shock, because that is just endotoxin is a really strong trigger for that. Let's get into some more specific diseases. Let's move on to the plague. There's 3 manifestations of the plague: the bubonic, the pneumonic, and the septicemic plague. So first of all, the bubonic plague, where we have development of buboes. So buboes are actually infected lymph nodes. So the infection is happening within the lymph node tissues, that the bacteria are taking up house there and multiplying within the lymph nodes themselves. And then as a result, the lymph nodes are swollen from the infection and the immune cells responding. And the lymph nodes can become so swollen, you can actually see them through the skin, like in this picture here. And that's a bubo. And so they can even become necrotic as a result of the infection. So bubonic plague is the least severe version of the plague. We're talking about 20 to 30% mortality rate with that one. Pneumonic plague is when it progresses into the lungs. And so pneumonic plague, you have the infection of this bacteria. This is a bacterial disease, and it's caused by one species of bacteria, Yersinia pestis, which is a gram-negative bacteria. And so gram-negative bacteria, they have endotoxin. And so we can also have endotoxic shock associated with this when we get the septicemic version of it. But the pneumonic plague is when the bacteria live in the lungs, and that can actually be spread from person to person. We don't need fleas, which most of us are aware that the plague is spread by flea bites. That was maintained within rodent populations, even in the dark ages of medieval times. Whenever they're having this disease, it was the fleas that were transmitting the disease. They were essentially biting the people and regurgitating bacteria into their bloodstreams. And then it would take up house in the lymph nodes, or could spread into the respiratory system and lead to the pneumonic plague. Again, pneumonic plague can be spread from person to person by aerosol and things like that. And then we have the septicemic plague, where we have actual infection within the bloodstream. And this can lead to intravascular coagulation, and we can have subcutaneous hemorrhage. So underneath the skin, having development of petechiae as well as purpura, which is a bruise-like effect. And then that can degenerate into necrosis and even gangrene, where the bacteria are now infecting the tissue and eating away at the dead and dying tissue as a result of the inflammatory process and the bacterial toxins and things like that. So we call this the Black Death. The Black Death is associated with the septicemic version of the plague, not all versions of the plague. So the septicemic version, the reason that's called the Black Death is because in the extremities, because of the necrosis and the inflammation resulting from the infection, would cause like the extremities to turn black, quite literally. And so fingertips, even full limbs might eventually suffer from this effect. And so that was the Black Death. If we are able to treat your septicemic plague with antibiotics, it's still a 30 to 50% mortality rate. This is with everything that we have that we can throw at you. And so that's your highest chances of survival. 30 to 50% mortality rate with treatment. But if without treatment, most septicemia without treatment will lead to death. Almost 100% fatality rate for most septicemia, because once it's actually able to multiply in the blood, your immune system is not going to get rid of it. And so it wasn't containing it in the first place, so the likelihood that it's going to clear it out is extremely low. So once septicemia sets in for most diseases, you're at a risk for mortality. All right, we just have the pictures. And again, Yersinia pestis, gram-negative bacteria that's associated with the plague. We still have the plague here today in the world. It hasn't been eradicated or anything. There's no vaccinations for it or anything, but we've just been able to control fleas and rodent populations and things like that. But we still have had cases in the United States. These are all of the cases from 1970 to 2017. So we're looking at like a 40 to 50 year period of time. And this is all of the cases. And so this is very rare to get it. And it is associated with prairie dog population. So prairie dogs are maintaining the plague in their own, they are rodents and they're maintaining it in their populations. And people that go hiking and stuff like that might be bitten by fleas that have this bacteria in them. And so we see this primarily in the Four Corners area of the United States. That's Utah, Colorado, New Mexico, and Arizona. And again, this is it's like one of those extremely deadly diseases that you don't want to get and can be difficult to treat it adequately. So if you are hiking in any of these regions, just be sure you're taking precautions against ticks and fleas, things like that, wearing long pants, maybe insect repellents and things like that. Moving on to infectious mononucleosis. Mono, also known as the kissing disease. This is caused by a virus called Epstein-Barr virus. It is a herpes virus. So again, all herpes viruses, they're forever. And so once you've had it, you have it, and it stays inside of your body. It is a DNA virus that incorporates its DNA into your DNA. And these guys primarily infect monocytes, including your lymphocytes. And so that's why whenever you get mono, you get those swollen lymph nodes, because your actual lymphocytes have gotten infected, and they're also simultaneously trying to fight the infection and keep it under control. Mono, if you haven't had it, or you think you haven't had it, you probably have. Over 90% of the world has had mono at some point. But during the teen years and like the younger years of life, if you're exposed to it, only it says 30 to 77%, which I think is a massive range. It just depends on the population. But 30 to 70% of the time, it will result in disease. So that means that the other side, which is 70 to 23% of the time, it doesn't cause disease. And so when I was in college, my boyfriend got mono, and he was hit hard. Like, it was rough. It makes you super tired. It's like having the flu, but worse, and it lasts way longer than the flu. So, and it makes you more sore even than the flu does. And so he got mono, and I was just waiting, man. I was just like, man, I'm going to get mono now too. He's my boyfriend. I'm going to get it. And I never did. But I went to the doctor, and the doctor did a blood test. He's like, you've got antibodies against mono. You've probably had it before. And I was like, I don't remember having the illness. He's like, yeah, a lot of people got it, and they never had the illness. And so I have it somewhere in my body, hiding out in me, just waiting, just like my chicken pox is. Maybe it can reactivate. And so some people actually do have reactivation mono, where it can like come back out again, just like shingles does, or just like herpes does. And so, herpes comes out with stress. And so some people can actually have mono come out with stress as well. And they can actually cycle pretty regularly with that. Other people, their immune systems just keep it under control. It's a matter of how your immune system responded to it and took care of it. So, but yeah, so once you have it forever. And it inserts itself into your DNA, and this can actually interfere with like your cells being able to go through the cell cycle, mitosis and everything like that. And so this can lead to development of cancer because that cycle can become unregulated. And so the cancer that's associated with infectious mononucleosis is called Burkitt's lymphoma. So it's a lymphoma. It happens with your lymphatic cells because those are the ones that were affected by the monovirus, the Epstein-Barr virus. And so your immune system keeps that under control if you have a healthy immune system. But people with HIV that develop into AIDS, their immune system is dysregulated, and so they can't keep it under control anymore. And so they're more likely to have presentation with Burkitt's lymphoma, which is one of the signs that somebody might actually have AIDS, which we'll talk about HIV in a little bit. All right, anthrax. We talked about anthrax in the skin chapter, but anthrax can pretty much infect most tissues of the body. And it's caused by a gram-positive endospore-forming bacteria called Bacillus anthracis. It is a zoonotic disease of herbivores, and livestock primarily can harbor this, and then it gets into the soil, and the endospores can stay there for decades. If it gets in the skin, it's cutaneous. If it gets in the lung, it's pulmonary. If it gets in the GI tract, it's gastrointestinal anthrax. And if it gets in the nervous system, it can cause anthrax meningitis. So here's the guy. Cool. All right. Hemorrhagic fever diseases, they are what they sound like. So if you get an actual fever, you have a raise in the body temperature. That's the fever side of it. And then the hemorrhagic side, you have hemorrhage. You're bleeding out. And so what happens with these guys is you have fragility of the blood vessels, and particularly in the capillaries first, and then it can even cause the larger blood vessels to become fragile and be more likely to leak their contents into the tissues and even then out of the orifices of the body. So these are extreme fevers, usually very high fevers, and then internal hemorrhaging will come along with it. A lot of these guys are caused by viruses, and they're like some of them are distributed by mosquitoes, and they're associated with the Aedes mosquitoes. And we see an increase of these moving further and further north as the global warming is happening. Further north, we're having higher temperatures that can actually harbor the mosquitoes that are carrying these diseases. So these three diseases are associated with Aedes mosquitoes, which we do have in the United States, the mosquitoes. We don't have yellow fever in the United States. This is found mostly in Africa and South America, and it's more frequent in like tropical climates where we have high mosquito populations. Then we have dengue fever. Dengue is endemic to Southeast Asia and Africa. We have had several epidemics. Now it has become relatively endemic to South and Central America and even seen in the Caribbean. We've had a few cases of dengue fever here in the United States, in Texas and Florida. It seems to be sporadic and not so much that it's becoming endemic here yet, but it likely will as the mosquito populations continue to multiply. Then there's chikungunya virus, which you probably haven't really heard of. It usually causes a pretty mild disease in people. It can cause pretty severe muscle aches, and it can even cause a very long-term, like painful muscle and neurological side effects. But chikungunya was originally found in Africa, and then it had moved to Central America, and in 2013 into the United States, and in Europe in 2014. So we do have this endemic now in the United States. It's commonly seen, again, in the southern states like Texas and Florida, and we do have outbreaks of it. It went from 0 to over 1 million cases in the Americas in just 3 years. And so mosquitoes can harbor this disease, and it can be hemorrhagic, and that is one of the kind of calling signs of this guy. It's not going to be as severe as like Ebola or something like that, but it can be associated with that. Dengue, something I forgot to mention, dengue fever. Again, not super, super common to have a hemorrhagic version of that disease, but it is one of the like hallmarks of that disease. Dengue more commonly is going to cause a very, very painful fever and severe high fever as well that can easily come over like 105 degrees Fahrenheit. And so that can be deadly from the fever. But dengue is also known as breakbone fever. And so the chills that are associated with this hemorrhagic fever, again, we're having an issue within the bloodstream. It's being systemic, and the inflammatory response to that is as well. And so the fever can actually cause chills that are so severe that can literally break people's bones. It's very serious. All right, then we have these hemorrhagic fevers that are not transmitted by mosquitoes, but are just transmitted by blood contact, typically, or associated in the case of Lassa fever. Lassa fever is maintained in multi-mammate rat, which I don't know really anything about. It's in Western Africa. But with Ebola, the one that we're kind of more familiar with, this is a true hemorrhagic fever that is spread through blood, and it has massive internal hemorrhaging associated with it. We're talking 70 to 90% mortality rate with these guys, and there's not much we can do for treatment for them either. Antivirals usually don't work fast enough to contain it once we see the signs and symptoms coming, and it usually kills people pretty quickly as well. But the good thing about like those kinds of hemorrhagic fevers is that in developed countries, we have enough awareness of hygiene that we're not usually, trying to roll around in someone's blood when they're hemorrhaging out. And so we can usually, it's a self-limiting infection because of societal norms within developing countries. We think Ebola and Marburg, which is very closely related to Ebola, it's basically the same kind of disease, that these are maintained in bat populations in Africa, and that they might be spreading it to like primates or other animals in like the jungles, and that whenever tribes that are in sub-Saharan Africa are hunting for meat to feed their families, that they are hunting bush meat like primates, things like that, that might actually be infected with Ebola, and that's how the infections almost always start. And then they need help. Their whole tribe is getting Ebola, so then people come in from the cities, and they could take this, the infection back out with them before they realize that it's Ebola, and it's harder to contain. And that's how we get outbreaks outside of those. But it's almost always originating from these smaller, more secluded villages that don't have as many hygiene practices or awareness of that. So that's usually where the problem is. With Lassa fever, it's also a very severe hemorrhagic fever, but you only see symptoms of that in 20% of the people that get infected. 80% of them aren't going to have any symptoms. So it's either you have no symptoms or you're going to bleed out to death. But only 20% of the people are going to have that. I'm moving on to the non-hemorrhagic fevers. So these are characterized by very high fever, but they don't have hemorrhage typically associated with them. One of these guys that we'll talk about at the very end can have hemorrhage in very severe cases, but it's just not as common as with the hemorrhagic fevers. All right, so most of these guys are caused by bacteria, and one of them is caused by a protozoa. Lyme disease is one of the bacterial diseases. It was discovered in Connecticut, and we really only see this in the northeastern portion of America. It is transmitted by tick bite, and it causes a very characteristic bullseye rash to develop most of the time. Some people can actually get Lyme disease without this rash, but more commonly, it's more that they probably missed it or didn't identify it or didn't recognize that until like after, like they maybe thought about it after the fact, like, oh, maybe I did have that or whatever. But this is a very slow progressing disease. You start off with fever, headache, stiff neck, and dizziness. So maybe some mild symptoms of something like meningitis, but again, it's very slow to progress, and it's not very severe, but can eventually, as time goes on, progress to cardiac and neurological symptoms, and even lead to a crippling arthritis if it isn't treated. This is caused by the bacteria Borrelia burgdorferi. It lives inside of cells. It's a spirochete, so it has a spiral shape. And it attaches to host cells and can actually move into the cells and live inside of the cells. It takes a long-term antibiotic treatment to treat this, but it is treatable. It's just usually misdiagnosed. Again, transmitted by tick bites. You know how that goes. Maintained in animal populations and transmitted to people that way again. It's mostly in the northeastern United States. Whereas ehrlichiosis and anaplasmosis, also bacteria transmitted by ticks. These are transmitted by hard ticks. We call them Ixodes ticks. These cause you feel sick. That's what we call febrile state. You're showing like the typical you feel sick symptoms. Maybe it's fever, your headache, you're achy. You might have the rigors, which is from the shivering, from the chills, from the fever. And most people will recover pretty rapidly without lasting effects. So you'll be sick for a few days from this and your own immune system will fight this off. So you don't usually have lasting illness from these. This is, again, bacteria, Ehrlichia chiefensis and Anaplasma phagocytophilium. Cool. Babesiosis causes a very, very similar disease, but it is different because it is caused by a protozoa. So ehrlichiosis, anaplasmosis, and babesiosis, all very, very similar signs and symptoms. If you need treatment from these guys, we'll give you treatment for all of it, basically. We're not necessarily going to test you for ehrlichiosis or anaplasmosis or babesiosis. We'll treat you for all three. And so we'll usually throw anti-protozoal drugs and antibiotics at you in order to treat the possibility that you have one of these if you're not recovering on your own. But again, most of these you're going to recover on your own. All right, finally, we have spotted fever rickettsiosis. I'm just particularly talking about Rocky Mountain spotted fever. And this is spread throughout the United States, Canada, and Central and South America. It's caused, again, it's transmitted by tick bites. And this is caused by bacteria Rickettsia rickettsii. And the bacteria causes an infection that can lead to chills, fever, headache, and muscular pain. Again, febrile state. You feel sick at first. But then 2 to 4 days after your initial symptoms, remember, initial symptom time period is called the prodrome. So 2 to 4 days after the prodrome, you'll develop the distinctive rash. And here's a picture of the rash. And so it's a petechial rash. We have bursting of blood vessels underneath the skin. It's a widespread rash all over the body. The lesions can merge and then become necrotic from the damage that's being done to the tissues from that inflammatory response. It causes cardiovascular disruption, hypotension, thrombosis, and hemorrhage relating to that cardiovascular disruption. It does have a 20% mortality rate if it is untreated. You can recover from this naturally, but it's going to take a long time and you're going to be very uncomfortable. Most of the time, the signs and symptoms are going to last for months. I mean, months. So you don't recover from this in a week. It's something that you will be miserable for months. I had a student in my Monday morning class that her husband or her father, somebody was related to her, family member, got Rocky Mountain spotted fever, and it was misdiagnosed for a very long time. Kept going to the doctor and saying he was having such and such symptoms. He couldn't move. He was bedridden. He couldn't work anymore, and was in a lot of pain for 7 months. For 7 months, and then finally the doctors were able to diagnose it as Rocky Mountain spotted fever, and he was able to get treatment and recover. You're going to treat it with antibiotics. It's a bacteria. Again, 20% mortality if it is not treated, but it's still 5 to 10% mortality with the treatment. So there's still risk for death there because it can potentially lead to hemorrhage and issues associated with hemorrhage leading to death. All righty, moving on to Chagas disease, a blood-borne illness. These guys multiply in the blood. They are protozoa called Trypanosoma cruzi. We looked at Trypanosoma in our microscopes. They're the little snake-looking guys swimming around with our blood cells. Yeah, so that's what they looked like. The ones we looked at in the lab were Trypanosoma gambiense. We're not really talking about that one in this class, but it causes African sleeping sickness. And like people get so tired from that illness, they literally just fall asleep driving and doing normal things. They can't, it's like narcolepsy, but like it gets worse and worse and worse and worse. And it can even cause exploding heart disease, which isn't like as explosive as it sounds, but the inflammation in the heart makes it so the heart can't function anymore. And the damage from that can lead to death as well. But with Chagas disease caused by Trypanosoma cruzi, again, a protozoa, and it's transmitted by this bug. These bugs are about this big. They're like this size. All right. So they're not super small. They're like the size of like an adult cockroach or something. And they look like this. And these are called triatamine bugs, but we also call them kissing bugs or assassin bugs. So if you see a bug like this, these are very common in Oklahoma. And the ones that we have in Oklahoma they very rarely transmit this disease, but it's much more common the further south you go, the disease is. So these bugs like to live in your houses. They like to live in the walls of your houses. They like the warmth and the nice stable weather and all that stuff. So they will actually live in your houses. In every apartment that I've lived in, I live in a house now and I haven't seen one of these yet, but in every apartment I lived in, I found at least one of these. So these are all over the place. You usually don't see them, but they are biting insects and they will bite you, and they're attracted to like the moisture of the eyes and the mouth. So they usually bite near the eyes or the mouth and they lap up the blood. And while they're lapping up the blood, they defecate. And so you've seen some of these things on like TV, right? Where they'll zoom in on the mosquito or the bug or whatever, and it like poops out liquid while it's drinking the blood. When it poops out the liquid, it poops out these protozoa with it. And so they carry that in their gut, it comes out in their feces, and then the protozoa can move in through the skin and get into the bloodstream that way. This site where the protozoa enter, it causes a massive inflammation from the inflammatory response. That site is called a shagoma. And so Chagas disease is characterized by the shagoma, as well as, when you have the acute version of the disease, mild to severe fever, nausea, and fatigue. If you have the chronic version that develops in your body, again, to your immune response and how it responds to it, you might have inflammation and disruption of the heart, the cardiovascular system overall, and the brain and the intestinal tract. And this can be deadly in some cases. We usually don't see this too much in adults because adults are usually disturbed awake whenever a bug is crawling on them when they're sleeping, but babies don't really get awakened from that sort of a thing. So we see this more commonly in babies and little toddlers. And so they will get bitten. And so precautions can be put in place to prevent bugs from being able to crawl up the sides of cribs and things like that. This is going to be more common again in the southern states, but these bugs do exist here in Oklahoma and they're not uncommon. They're less likely to carry the disease though. Moving on to malaria. Malaria is another protozoal disease. The disease of malaria, basically we have a fluctuation of the signs and symptoms. They'll come in periods of 48 to 72 hours, 48 to 72 hours of signs and symptoms, 48 to 72 hours of no signs and symptoms, and then cycling through that just forever. And so what happens is the signs and symptoms include malaise, fatigue, aches, and nausea with or without diarrhea, as well as chills, fever, and sweating. So you have massive fever and then these general feelings of unwellness with that, and that will cycle on and off every 42 to 72 hours. If you have this falciparum version of malaria, which is more common, you also might have a persistent cough as well as weakness that constantly lasts. Those won't cycle. And so cerebral malaria is when it moves into the brain and it starts affecting the blood vessels of the brain. In the blood vessels of the brain, those capillaries can become impeded by the protozoa, basically, and we don't get proper blood flow to the brain tissue, and that will lead to coma and death as a result. Protozoa, they live inside of your actual blood cells as well as in your liver, and so they can cycle between the liver phase and the blood cell phase, and that cycling, when they're bursting out of your blood cells, when they're multiplying, that's what causes this fluctuation of the signs and symptoms. This is caused by an apicomplexan. And so this is a term, apicomplexan is a term that we use to describe protozoa that are non-motile. And so we have protozoa, if we remember back to chapter 5 really quickly, protozoa are basically single-celled eukaryotic organisms that are capable of being motile either by flagella, cilia, or by amoeboid motion, which we call pseudopodia. And so if they can move, they move like that. But the ones that can't move, we call them apicomplexans. Apicomplexans are all non-motile, and they're also all parasites. So they all cause, they're true pathogens. They all cause disease in animals of some kind. So this, for malaria, the apicomplexan that causes malaria is named Plasmodium. That's the genus name. And then for the specific epithet, which is the second part, we'll remember binomial nomenclature, we would call it Plasmodium falciparum is the species name. Falciparum would be the specific epithet of that. That's the more common one, as well as Plasmodium malariae and vivax. Depends on what part of the world you're in that you're likely to see which of these malarias it is. But yeah, if you're interested in the life cycle of these guys, they do actually multiply within the gut of the mosquito, and that's part of their process of their life cycle. So the mosquitoes are actually infected with the malaria, and they grow in the gut of the mosquitoes. And then those versions, after they mature in the gut of the mosquito, get passed into the people and then cycle within the people. So again, it invades the blood cells, and whenever they burst out of the blood cells, it releases fever-inducing chemicals. It causes severe anemia from the bursting open of the red blood cells, and that will cause spleen enlargement as well as liver enlargement, because these guys also infect the liver cells as part of their life cycle. They are able to vary the antigens on the surface of the protozoa so that your immune system can't latch on. It can't like create a proper response to it. So that also makes it really hard to develop vaccines for these guys. So there are vaccines being developed, and it seems to show promise. We recommend it for children in endemic areas. The problem is that it requires boosters. And so I don't know if you know, but sub-Saharan Africa is not exactly friendly to people like going to see doctors to get their booster vaccines regularly, right? Talk about people that live in underdeveloped countries that they're likely to even see a doctor ever. And so the likelihood of it actually being effective in those endemic regions is actually pretty low because of the reliance on the booster vaccines. So the best bet for controlling malaria is through mosquito control. And so mosquito abatement, so lowering the mosquito populations, we can do that by eliminating standing water near populations, spraying insecticides in the area, and introducing sterile male mosquitoes. So we can genetically manipulate male mosquitoes to be sterile, and the female mosquitoes only mate once, and then they will die. So they'll usually mate with the male and then go lay their eggs and then die. But that mating process like triggers that whole response. And so we can have them mate with sterile males, and the female mosquitoes will die as a response from that. So we introduce these sterile males into these areas where this is endemic in the hopes of lowering the populations. And of course, to protect the people, we recommend using mosquito nets, especially in the sleeping areas and the living areas, as well as mosquito repellents. If you're traveling to an area that is endemic for malaria, we recommend that you take chemoprophylaxis. And so this is taking anti-malaria drugs before you go to the area so that malaria can't take hold and reproduce in your body. Yes, it's the same thing with the picture. All right, moving on to HIV. HIV stands for human immunodeficiency virus. It was isolated in the 1980s. It was originally associated with immunodeficiency, and that's how we even knew that it existed. We had populations of people that were having these really strange opportunistic infections that we weren't seeing in the rest of the population. And it seemed to be localized within the gay male population. And so when it was first identified, we called it GRID. It was called gay-related immunodeficiency. But now we have learned from studying the virus, this is not a gay-related disease, this is a human-related disease. And so this is spread within the blood and other body fluids. Semen and breast milk can spread the virus, but there is not enough viral load in saliva or sweat or urine for it to be transmitted those ways. And even with semen transmission, being in contact with semen wouldn't be enough to transmit it. It would need to be able to have an opening to the blood. And so even with sexual contact, you usually have micro tears that are involved. You might not have blood showing, but micro tears maybe in the vagina or even in the anus would open up, opening into the bloodstream for the virus to get in. So that's where the issue is really coming from. It's caused by a virus that falls into the family retrovirus. And retroviruses are RNA viruses that can like basically turn their RNA into DNA, and then it inserts that DNA into the genome, which we've been talking about for a while now. The complex of symptoms that's associated with AIDS is going to be having pneumonia caused by pneumocystitis gyrovecii, which is extremely uncommon. We don't see this very often. So pneumocystitis gyrovecii is not something that the healthy, people with normal immune systems would ever be getting. Kaposi sarcoma, another cancer very similar to like what we see with Burkitt's lymphoma. It's usually kept in control by our immune systems. Sudden weight loss, swollen lymph nodes, and general loss of immune function, all related to having AIDS. And so we don't really consider it AIDS until their T helper cells, their T cell count, falls below 200 cells per milliliter. That would be considered very low, and that you would be having a decreased immune response. And that's how we define AIDS, when your T cell count is below 200 cells per milliliter. Most people have more than 500 cells per milliliter. And so we're going to define it by the count of the CD4 positive T helper cells that are your captains in your immune response. So signs and symptoms associated with HIV infection and AIDS. When you have just HIV, your initial signs and symptoms will be fatigue, diarrhea, weight loss, and neurological changes just from having the virus itself. but it's usually pretty mild. Like the virus, if you weren't getting other infections from the immune response being dampened, having the virus itself doesn't usually cause that many severe symptoms, and you could live with it just fine. But the problem is with the opportunistic infections that are happening as a result of your immune system being deregulated. And so this is going to be so you'll see immune system deregulation, and you'll see problems with hormones and metabolic disturbances as well, being affected by the fact that all of your captains aren't able to control infections and aren't regulating things the way they're supposed to. And so that's usually where a lot of these symptoms are coming from, is from the dysregulation of the immune system. You also see lesions on the brain, meninges, spinal column, and peripheral nerves. And again, all of this is probably due to these weird opportunistic infections that most people aren't going to be getting from normal, just environmental bacteria. There's two different versions of HIV. HIV-1 is by far the most common. And again, this is an RNA virus that turns its RNA into DNA and then inserts its DNA into the CD4 positive T cells. And then those T cells can crank out virus whenever the virus wants it to, basically. It is highly closely related to simian immunodeficiency virus. So we believe that this basically mutated from that and moved into the human population. Anyways, interesting fact that I feel is kind of horrifying. If you take the entire population of South Africa and look at all of the people who are of age to be sexually active, so we're talking 16 and above, up to like 45 years old is what the population that we're looking at. 50% of those people have HIV. 50%. 1 out of every 2 people has HIV in South Africa. So if we think this isn't a big problem, if we think it's something that we're keeping under control in the United States, I have some interesting figures and facts for that coming up too. So again, this is just reviewing what happens. The RNA gets in, it makes a DNA copy of itself, it moves that into the nucleus, it gets incorporated into the actual host genes, and now it can make mRNA from that and make new viruses going on from there. It's transmitted, again, through blood, typically. So it's usually trauma and needle sharing that is the most common, as well as sexual transmission. So unprotected sex is a very common cause of spread of this, and it can be transmitted through the breast milk as well. There's been evidence that's been shown that babies can be born free of HIV, and when they feed on breast milk from somebody that is HIV positive, that can spread virus to the babies. So it is actually recommended that if you are a mother and you have HIV, you would stay maybe on your drugs to keep your viral loads down, and you can have a healthy baby without HIV. That's entirely possible now with the medications that we have, but you would probably not want to breastfeed just to decrease the risk of passing that to the baby. If you weren't able to stay on the drugs during that time. So anyways, urine, tears, sweat, and saliva are not considered sources of infection typically. Again, if you are a user, we'll talk a little bit about preventing it in a minute, but again, so 50% of South Africa, and there's plenty of other countries that are racking right up close to South Africa there. It's more common in Africa than it is in other parts of the world. 30 million people, it's estimated that more than 30 million people have died from HIV-related disease, so AIDS and what it can lead to. Since the 80s, 600 million people have become infected, and some of the people have died off. So now we have 38 million people infected currently worldwide. 1.2 million infections of those are currently in the US. 1.2 million people have HIV, and in the US, based on our estimates of what we have done on our populations, about 15% of the people in the US who have HIV don't even know they have HIV. And so you could be having sexual relations with somebody that doesn't even know that they have HIV, and they might tell you that they don't have, be sure that you're getting tested when you get new sexual partners. Just be sure that everybody's getting tested, please. You can get diagnosed as having HIV, and you're considered HIV positive for life, no matter what, even if your viral loads are undetectable. That just means that the vial of blood that I took from you didn't have the virus in it, but you might still have a virus floating around somewhere else. It's just undetectable from the machine. So you're considered HIV positive for life for now. And having HIV doesn't mean you have AIDS, but if you don't treat the HIV and keep the viral load under control, then the CD4 count goes down, and then you have AIDS. So the way that we detect the virus is through ELISA, and antibody tests typically is what we're going to be doing. So we look for antibody and not the virus, because you might not find the virus in the blood, but if a person has antibodies to HIV, they have HIV. So there's that to look for. Nobody clears it out of their body, right? Best ways to prevent HIV: avoid sexual contact with infected people and use barrier protection. That's always a great option. So if you have a new sexual partner, until everybody can get tested, use barrier protection and be diligent about it. Do not share needles with anybody if you are, engaging in illegal drug use. Don't be sharing your needles with people. But if you do share needles, and maybe even if you had patients, because you're going to have patients that might abuse drugs like that and might share needles, inform them that they can bleach their needles, and then their needles are safe to share. Not that I'm condoning drug use, intravenous drug use, but apparently you can bleach the needles and it will eliminate these blood infections like hepatitis and HIV. HIV positive individuals with uninfected partners should begin a regimen of antiretroviral drugs so that they don't pass on the HIV, and their uninfected partners, we now have PrEP and STEP that are used for preventing the spread of HIV. So the uninfected partner can take the drug like prophylactically to prevent the spread of it. And it seems to be very effective with that, but I still highly recommend, even if you have a partner that has HIV, use those medications to prevent it, but also use barrier techniques. Just to be safe, because once you have HIV, it is a lifelong infection and it's very expensive to control it. And it requires diligence. There are vaccines that we are trying to develop, but the problem is viruses mutate very quickly. And just due to the nature of the spike molecules on HIV, it's difficult. So treatment usually should begin immediately after HIV diagnosis, so we can make sure that your immune system stays strong and doesn't get killed from the HIV infection. Antiviral chemotherapy and other drugs will be used to treat the opportunistic infections. We usually treat with things to inhibit the reverse transcriptase, which is the best target, because we don't have anything like reverse transcriptase in our bodies. We can't convert RNA back into DNA naturally, but only HIV can. And that reverse transcriptase is the best target for the drugs. The other molecules involved in incorporating that DNA into the nucleus, they're also great targets, as well as protease, which, like HIV's protein, is made in one big old long strand, and then protease cleaves it into the parts that it needs to use. And so if we can inhibit protease, then we can inhibit it from making its proteins as well. So those are great targets for that. All right, our questions. All right, are we ready? You guys are ready to participate? Okay. What disease does Yersinia pestis cause? Plague. Excellent. What organism causes Lyme disease? Ooh, yeah. Yes, close, right? So we definitely, you guys will have choices on the exam. Borrelia burgdorferi for that one. And what type of organism is that? Do you guys remember? Bacteria, exactly. How is malaria transmitted? Like, what transmits it? Mosquitoes. What type of organism causes malaria? Protozoa. Excellent. And finally, what does HIV use reverse transcriptase for? Right. So to turn its RNA into DNA, and then it'll put that DNA into our genome. There's a little video on here. I'm not going to play it, so we can just go to lab early today. But if you guys wanted to watch it and learn a little bit about this, then that's there for you to watch. All righty, let's head to lab and do our very, very last lab. I'm sure we're all going to cry a little tear over it. You won't ever have to open that stinky incubator. It'll be tears of joy, right? Tears of joy. No more stinky incubators. All righty.